I mean, we're taking innovation, and we are absolutely applying the best technologies in the world to these issues that Bob just described. Most importantly, we're continuing to try to do everything in our power to inspire the best minds that I've ever met in my life to keep innovating around the things that we're doing. The next step of this opportunity is to invite someone to the stage that's going to be, for me, probably one of the most moving opportunities in my career. I got the chance to meet Nancy Frates. This was back maybe a month and a half ago. My daughter went to Boston College, and we all knew Pete's story of ALS and how that all happened. And I got a chance to truly meet the person and what's behind the family, the Frates family, and what they've been able to achieve in raising over $250 million for ALS. And they've done this, they've looked adversity in the eyes, and they said, let's take it on as a family and as a complete community, and it just spread around the world. So what I would like to do, we're going to have a panel come up after Nancy makes her comments, but I'd like to introduce to you Nancy Frates, who is on a mission with her family to defeat ALS. She's a proud mom of Pete, ALS patient advocate and inspiration for the ALS Ice Bucket Challenge, which has raised, as I mentioned, $250 million. She and her family have found their sense of purpose in working tirelessly to raise awareness around ALS and to stimulate funding for research. It's been 75 years since Lou Gehrig's delivered his famous farewell speech, and the only new drug and no new cure has been found. This, says Nancy Frates, is unacceptable. She did a TED Talk back, I don't know, maybe a year or so ago. It's had over 1 million viewers. And the reason is that this is where the rubber meets the road. You can sit there and crumble when you are brought to your knees as a family around any issue that you might have. And health brings us to our knees, as we all experience in our lifetimes. Nancy and her family and Pete are just angels trying to do something about ALS, which is just a devastating disease. So, Nancy, thank God you're here, Nancy. Thank you. You're awesome. Thank you.
Thank you. Good morning, everybody. Bob Coughlin, my hat off to you. I couldn't have said what you said any better. Amazing. So the first thing I'd like to do is just give you a quick overview of our story so I can kind of bring you up to date and then really dovetail it all to what you all really smart people are doing here. I'm probably the person with the least amount of letters after their name that is in this room. So our family was just like your next door neighbor. We were empty nesters. We had 3 kids graduate college. One moved to Manhattan, one moved to South Boston, and one moved to Charlestown. Pretty much what happens here in Boston. And it was my middle son, Pete. When he was a little boy, he had a traumatic birth, as a matter of fact. He got staph infection as an infant. He was born the day before New Year's Eve, and at 11:45 on December 31st, 1984, I had 2 pediatricians walk in my room and tell me how sick my son was. He was less than 24 hours old, and he fought that infection off. It was in his blood system, and he fought that infection off, and I look back on those days and I think to myself, number one, thank God for really smart doctors and people who care and come in, left their New Year's Eve party with their family and friends to come in and take care of my little boy. But number two, I saw a kid with grit and determination. This kid was chosen. He was chosen at birth. And as he went through his life, he was a leader. He was a servant leader. He was the best teammate you could ever have. Any of his friends will tell you, any of his teammates and classmates, they always looked up to him, not because he was the best player on the field or the smartest kid in the classroom, but he was the one who was trying to make them be the best they could be. That's the ultimate teammate. And as he went into high school and college, he really excelled in sports. He played football, hockey, and baseball 4 years at St. John's Prep up on the North Shore, and then was recruited to Boston College to play baseball in the ACC. He had a tremendous year, graduated BC. MLB didn't come a-calling, unfortunately, so he went to Europe and played in Europe and made friends and teammates all over Europe as he played baseball. And then, as I said, he came home, moved to South Boston, had a beautiful girlfriend and a job. And in August of 2011, I brought my family down to Florida. My daughter had just gotten married, and I said, I want that one last vacation before there are all these other people that are gonna infiltrate into our family, right? And during that vacation, Pete told me that he was gonna quit his job. And I remember thinking to myself, okay, you live in South Boston, you have school loans. Okay, why are you gonna quit your job? And he tells me that he wasn't doing what he was supposed to be doing. Mom, I'm not passionate about what I'm doing. I'm supposed to be doing something else. And then he went back home. After we left Florida, we came back to Boston, and Pete was still playing baseball in the Intercity League in Boston. And during the championship game, Pete was up at the bat and got hit in the wrist, and his wrist went limp. And for the next 6 months, he said, don't worry about it, Mom. I'm going to the doctors. They're gonna find out what's wrong with me. And on March 13th, 2012, we went in to one of the most amazing hospitals in the world, right here in Boston, went into a neurologist's office, sat down, and thought they were gonna tell us he had a pinched nerve. And the doctor looked at us, an amazing doctor, a compassionate doctor. Believe me, in the last 5.5 years, I know that determination now. An amazing person who sat there and took his hand and said, Pete, we've put you through a lot of testing. Now we know it's not Lyme disease. We know it's not sprain. We know it's not infection. We know it's not MS. We know it's not lupus. And to this day, I remember seeing that hand go up and go up. And I'm thinking to myself, where is this guy going? And then he put his hands on his knees and he looked right at Pete, 27 years old, and says, I don't know how to tell you this. You have ALS. Well, being a parent, my whole world changed in a second. Not only just because now I was dealing with my child being sick, right? It doesn't matter where on that level of sick he is. But you know what? I didn't know on the barometer of sick how sick he really was, because I didn't know what ALS was. I knew it was bad, but I didn't know the context of what bad really meant. And I knew Lou Gehrig was 75 years ago. We were a baseball family. Well, 75 years, come on. There's a lot of technology and improvements and innovations, and oh yeah, this thing called the internet, right? It's gotta be something. And then the second shoe dropped when that amazing doctor looked at us and said, Mr. and Mrs. Frates, I'm sorry to tell you, there's no treatment, no cure. Pete has 2 to 5 years to live, and it's 100% fatal. And then as we left the diagnosis room and out to the receptionist with the doctor, I have to tell you, this was probably one of the worst moments in the journey. When we looked at the doctor and said, okay, well, when do you want to see him again? And he says, in 3 months. And I thought to myself, 3 months? You just told my 27-year-old kid he has 2 to 5 years to live. Let's put that in percentage of how long he has. Why don't you want to see him tomorrow? He's so sick. Mrs. Frates, I'm sorry, there's nothing we can do. And when he comes back in 3 months, we just want to check how far he's progressed. So that is our story. That was our journey. But our new journey started 6 hours after that was delivered. That's when my son, that servant leader, sat at a kitchen table with his sister and brother-in-law who came home from New York, and his other brother who came home from Charlestown, his mom and dad, and his girlfriend, who he had only been dating for 8 months and was a senior in college, came home, and we're all sitting around the table. And he looked right at us, and it's a word you've heard I don't know how many times in the 45 minutes I've been in this room already. The situation is unacceptable. If you come across a situation that's unacceptable, you have one of 2 choices. You can cower under it, or you can take it head on. And that's exactly what my son did. He looked at us and he gave us the charge. 6 hours after diagnosis, he said a bunch of stuff. But the things that I remember the most are, we're not going to look backward, we're going to look forward. We're going to change the trajectory of this disease. I'm going to get this disease in front of philanthropists such as Bill Gates. I'm going to get Major League Baseball to reignite its commitment to a disease named after one of its heroes. And then he said the words that I wake up to every morning and ring in my ear and put a smile on my face. What an opportunity we've been given to change the world. Now, I don't know about you, but when I was sitting there were a lot of words I would have used to describe that diagnosis. Opportunity wouldn't have been one of them. But I had a kid who had educated himself to the state of this disease, and he was determined to do something about it. So for the next 2.5 years, we continued with Pete's vision. We marketed and branded a disease. We educated a whole new demographic to a disease that was named after someone in 1939. Now, in August of 2014, Pete was already in a wheelchair. He had lost his ability to eat, and he had lost his ability to talk. So he was on a feeding tube and was using eye gazer technology to communicate. And because he's so present in his life, he identifies opportunity where others may have missed it. The Ice Bucket Challenge had been around for over a year prior to August of 2014. What's the difference? Pete saw it, saw the opportunity, got strategic, and got purposeful. And during the month of August 2014, we hoped that you would open your hearts to our story, and you did. Facebook has reported 17 million ALS Ice Bucket Challenge videos were posted during the month of August 2014, viewed by 440 million unique viewers over 10 billion times. So we asked, and YouTube has reported that 150 different countries posted ALS Ice Bucket Challenge videos. So you opened your heart to our story. Then we hope that you would open your mind to this disease, this unheard of, underfunded, and not included disease. And you did. Wikipedia between January 1st and July 31st, 2014, the ALS page was hit on an average of 164,000 times a month. August 2014, the ALS page was hit over 3 million times. So you opened up and you learned about this disease. Well, then we hoped you'd open your wallets. And as Kevin said, in 6 weeks, we raised over $225 million for a disease that was so grossly underfunded. But more importantly, we woke up the world to the reality of a disease that had been in the shadows too long. So I'd like to just put it in the context of precision health and talk a little bit about how important it is in our disease, but how challenging it is. Right now, I will tell you that the Ice Bucket Challenge funds are funding about 3 to 4 different collaborative big data precision medicine studies in our disease. Now, why is this so important, especially in our disease, and why is it so challenging? Well, the first is that when you turn and you look at that doctor and say, why does my kid have this disease? They can't give you a reason. Now, of course, one of the things is what you've been talking about a lot here, CTEs. Yes, Pete had concussions. He had one that was so bad that he had to be hospitalized for it. And you know what? Out of football, hockey, and baseball, it was in baseball that he got the worst concussion, which I think people, that takes him back a little bit. But in our disease also, when you're given the diagnosis, 10% of the people who get an ALS diagnosis pretty much know they're going to get it because it's familial, coming down in the families. And if it's in your family, it is devastating. It attacks 50% of a generation. I have seen familial charts with X's through all the people in their family that has died. But the other 90% of us, our diagnosis, you know what the term is? Sporadic. I didn't even know that was like a medical term. Sporadic. Mrs. Frates, he has sporadic ALS. Okay, so that's a challenge. We don't know why people get it. The diagnosis. Unfortunately, our disease is still a diagnosis of elimination. So that ladder climbing, right? That was the doctor saying, well, we tested this, we tested that, and just getting that. And then, of course, you talk about painful, invasive, expensive testing. Has anyone ever seen an EMG performed? It is absolutely devastating. And the other thing is that we only can conclude ALS post-mortem. So you have this diagnosis problem in that not only are the brightest minds who are in neurology, specifically in ALS, challenged to diagnose it, but I will tell you, I have found people throughout this country and talked to so many ALS patients that never get diagnosed or have surgery. I talk to people who have had their spines opened up, have had shoulder surgery when their arm went weak. So this awareness campaign that we have been on and that we continue on is not just for moms and dads and daughters and sons, but also for doctors, GPs, neurologists, to put this disease back on the table and take note of it. Another challenge is our clinical trials. Our clinical trials are just very, very hard and difficult to monitor and also to populate, because our disease, no two ALS patients are alike. When you get diagnosed with ALS and they tell you to come back in 3 months, they have no idea when you walk back in 3 months how far you've progressed, because different rates of progression. And you look at the doctor and you walk in and they're like, okay, well, we're going to test Pete to see what he can and what he can't do. And meanwhile, someone who was diagnosed on the exact same day is either better off, it hasn't progressed as much, or is worse. So in a clinical trial, to have cohorts, and that's why the precision model is so important in our disease, to try to track, get the same people, like people, to get these clinical trials to help with the data. Our measuring tools in ALS. And I'm a layperson. I am not a doctor. I'm not a clinician. I'm not a researcher. But from my point of view, from being a mother of a patient, these measuring tools, this ALS, FRS, it's so subjective that the data is not objective at all. This is the ALS, FRS. It's a questionnaire that the patient fills out. Now, let's think about this. You have a disease. I don't know if we have any ALS patients in this room, and if we do, I'm just talking to try to get the emphasis of what this is, what we go through. But you get a questionnaire as the patient. Okay, a rate from 1 to 5, your arm movement, your leg movement, your ability to speak. Now, try to get into the mind, and believe me, as a mother, I'm probably as close to the mind of an ALS patient as you can possibly get. But you know what the end game is here. So do you think when you're ranking yourself from 1 to 5 that you're going to put, where do you think you're going to rank? You're going to rank the least possible number that you can. It's just human nature. The other one is an FVC, forced vital capacity, which is unfortunately the terminal part of ALS, when they can no longer breathe anymore. They take, it looks like a kazoo. and puts them in their mouth, and they tell them to breathe into it so they can measure the forced vital capacity. Well, there are so many problems with that. The first being, how tight can someone put their mouth around this mechanism that kind of, looks, like I said, looks like a kazoo. And the other thing is that the person who's giving the test will look at the result and say, oh, you can do better than that. Let's do it again. So we do it again. I sat in a room once where they literally left the room for a half an hour and came back in and had Pete do it 3 more times. So our measuring tools are very, like I said, they're faulty at best. Care versus cure. I think that's a huge one in our world. The Ice Bucket Challenge was all about funding for a cure. Right now, we're battling the care. And like Bob was saying, the expense to have my son home right now. Pete has been on a ventilator for 3 years. He virtually has lost all muscle movement other than a little bit in his face. He calls himself the bionic man because he lives on machines. But the other point is as this disease progresses, I'll give you the visual that I went to bed with the night that Pete was diagnosed. Remember, now I'm really dating myself, back in the 80s, Pac-Man, the little yellow Pac-Man. I went to bed at night, and all I could think of was that little yellow Pac-Man rummaging through my son's body. And every morning I would wake up and wonder what Pac-Man took away from my son and what we were going to face that day. Because ALS patients grieve every day. Could it be the day that Pete walked in the house at 28 years old with a set of keys and did a mic drop with his keys, knowing he was never going to be able to drive a car again? So the problem, again, another challenging piece in our world is this preventive medicine, because these milestones now have to be met with treatments. But the patients meet these milestones with denial and anger. Catfish Hunter, the Hall of Fame baseball pitcher, he had ALS. I don't know if a lot of people know that. Did Catfish Hunter die of ALS? Well, I would say yes. But Catfish Hunter was losing his ability to walk. He didn't want to get in that wheelchair. I know what that is. My husband and my son would walk with Pete with his arms because Pete didn't want to go in the wheelchair because they know once they go in the wheelchair, they're never getting out. It's like a defeat for them. Well, Catfish Hunter fell down a flight of stairs and hit his head because he didn't want to give in to that milestone. He died a month after he fell from a brain bleed. And the other thing is that our symptoms end up mimicking other diseases. So Pete has an ALS doctor, he has a general practitioner, he has a urologist, he has a pulmonologist, he has an infectious disease doctor, he has a psychologist. And there are a bunch of problems that come with that, but the first one being is that when he needs a drug for a symptom, we didn't know which doctor to call. And then if we called the wrong doctor, they wouldn't prescribe it because it was out of the realm of what they're supposed to do. The other thing is that Pete, when he gets all this pneumonia and this infection, it mimics CF, like Bob's son. So Pete has been put on a CF drug. But because the FDA approved it for CF and not for ALS, it's considered open label. And it costs our family $3,000 a month to keep Pete on that drug, which we will gladly do. But I just want to take this last minute to talk about the good news and going forward. So funding, the Ice Bucket Challenge was a start, and it is continuing. During the last 2 years, good news, people like you, for data, we have 4 new genes, and the biomarkers are coming at a really swift pace right now. Are we collaborating? That was another incredibly unacceptable situation that we found ourselves in. We are collaborating like you can't believe right now. Project Mine and Dr. John Landers out at UMass Medical, when they found the gene, that was an international collaboration. We have a change of mindset in our disease, and this has a lot to do with precision medicine. People aren't dying from ALS anymore. They're living with ALS because we're using treatments for like CF, because we're educating people that you can live with assistive technologies and getting the right medications that will help with your symptoms. You can actually live with it. The discoveries, they are the result of a movement that was 2014. And in August of this year, we had our first effective treatment approved by the FDA, Radicava. Radicava, and I get myself in trouble sometimes, again, I'm not a doctor, but from what I understand, in early diagnosis, helps slows the progression down by 33%. Now, that might not be true. We're still working on the data of it, but I'll tell you this. Remember when I said the worst day of my life was when we walked to that receptionist and they told me not to come back for 3 months because there was nothing they could do? The ALS diagnosis right now should be, come back tomorrow. We have an infusion drug to put you on that has shown effectiveness in early diagnosis. And the disruption of the Ice Bucket Challenge. We disrupted the ALS space. We disrupted philanthropy. We disrupted social media. And we disrupted the FDA because that drug was approved in the United States without any US data. They used the clinical trial data from Japan. And Janet Woodcock told us at an advocacy meeting that she heard us. So I ask you, please hear us. Put ALS in your conversations, in your boardrooms, and on your lab bench. Disrupt. It is our time. Thank you for your work you do. I can't tell you how grateful patient advocates are for people like you. Thank you so much.
Awesome job. You're going to stay up here for the check. That was awesome. Nancy, obviously, you've opened up our hearts, and we've got the greatest minds in the world here today. If I could invite the panel group up to the stage.
They're in the bad seats.
They're in the bad seats. They're in timeout. Some of the biggest minds, if you could come up to the stage, please. I couldn't help but think as I was listening to Nancy, you guys might remember I came out of Steve Jobs last year because I was almost so embarrassed how far this technology came as opposed to what we do for health. We were so enthralled with this technology. We also then talked about there being 6 quarts of blood in our body, and there's 3 pints of cerebral spinal fluid. And there's a blood-brain barrier that separates them. And there are biomarkers circulating 60,000 miles through our body every minute. That's a treasure trove of information. And those biomarkers is what we're now looking at taking and putting into almost an iPhone kind of application where you look at our body and those veins as the internet, and then you have applications across every biomarker to see these diseases. When you ask the question, how many of you have ALS? I'm hoping there's no one here in the room. I'm wondering how many people that might have it and don't know it because we haven't applied these technologies, but we are. We're getting very close. So important. So what I'd like to do is introduce this distinguished group that are now looking for treatments. Today's panel is going to be more about looking for treatments and vaccines to prevent this, which requires the drugs that we are trying to get through the FDA, and there's a lot of challenge, obviously, with that. So you welcomed yesterday onto our panel Ramon-Diaz. I won't spend a lot of time on him, but famous neurologist, PhD researcher, as well as MD clinician in the area of brain health, focused a lot on concussions and a lot around that topic, but it's an environmental factor. We've decided concussions are an environmental factor that leads to many of the diseases that we have. So Ramon's on the panel. We've got Jeff Dage, a senior research advisor from Eli Lilly, which is really focused on some of the top neurological degenerative disease drugs in the world. He has over 20 years of experience in drug discovery and early clinical development, has developed new methods for purification and physiologically relevant human cell models for diabetes, toxicology, and neurodegenerative diseases, has continually been on the forefront of identification, development, application of these novel technologies. And so these biomarkers that we have circulating, we now know that we can tee them up and link them to the drugs so that the drugs won't be the fourth leading cause of death in the United States due to toxicity. They will actually be the thing that helps us get better and not get worse. Too much toxicity, not enough efficacy. The biomarker link-up that you're going to hear today is all about making all the efficacy occur without all that toxicity. Al Sandrock, he's the Chief Medical Officer of Biogen. I've gotten to know Al through so many other people that just keep saying to me, this is one of the smartest brains in the world, MD, PhD, around coming up with the next drug. And I got to tell you, I've had a chance to interact not just with Al, but his organization. He's got so much admiration from all of the researchers at Biogen, one of the most successful biotech companies in the world. They follow Al because Al is such an innovative leader of chemistry that's really leading to the next set of drugs. And I know he's got some breakout ways of thinking through this. And so soliciting his input on this panel is going to be very important. But before we get started, we're going to ask for a small keynote address from someone that just lit my life up when I got to meet her. This was a few months ago. Her name is Denise Faustman. She's an MD, PhD Badger. She comes out of the Midwest like myself. I got my High State Buckeye stuff here today for you. In fact, Denise, I'll go ahead and give you one right now. And Nancy, you can take one and pass them down. But she's a director of, she basically said Harvard looked at this and said, we got to get you from the Midwest into Harvard, because if you're going to figure out a way to prevent type 1 diabetes, we want it to be coming out of Boston, not out of the Midwest. So it's pretty amazing how they were able to pull her in. But she's director of immunobiology laboratories at Massachusetts General Hospital and associate professor of medicine at Harvard Medical School. Leads the most advanced clinical trials in type 1 diabetes, advancing the TNF boosting concept with phase 2 type 1 diabetic trials using a 100-year-old generic vaccine, BCG, a strong inducer of TNF and Treg cells. So it's amazing. She took this vaccine that's been around forever, and she's turned this thing into something that we are all going to hear about, which is amazing. She's awarded National Institutes of Health and National Library of Medicine, Changing the Face of Medicine Award, the Oprah Achievement Award for Top Health Breakthrough by Female Scientists, and the Woman in the Science World from American Medical Women's Association, and Wyeth Pharmaceutical Company. So without any further ado, Denise, it's so great to have you here.
Thank you.
Thank you very much.
I was sitting over on the side saying, these are tough acts to follow, okay? So I hope I can provide some entertainment as well as some data. So I'm going to talk about BCG today, just so nobody's asleep. It's not Boston Consulting Group, okay? You can't imagine how many times I have BCG on the program. They go, oh, you're going to talk about Boston Consulting Group. So BCG is the oldest and continuously used vaccine in the history of the world. Last year, 100 million infants were vaccinated with BCG. 4 billion people have received the vaccine, and it's been continuously used for about 120 years. So we're going to show you the value of this vaccine, not only in our data, but all the global trials going on as they relate to the immunomodulatory effects of this very, very old drug. How do I advance the slides? Did somebody steal the pointer? Okay. Oh, this is very high tech.
That green button.
Okay.
There you go.
Okay, so it's the oldest vaccine on the planet of the Earth. It was devised a long time ago. I'll talk to you about how high tech it is. It's the most low tech drug going to be spoken about in 2 days of this meeting. There's advanced global trials. There's 16 trials going on in Australia, US, Canada, and Europe, repurposing this vaccine for diverse forms of autoimmunity, for reversal of allergies, and also prevention of infections. We'll show you some of the scientific basis for using this. TB was the #1 killer at the turn of the century, but they realized that milkmaids never died of tuberculosis. And it was developed as a result of realizing these young girls who milk cows were exposed to another form of bacteria. So it's the avirulent form of TB. In the US, it's only licensed for one indication at very high dose bladder cancer. And if you look at the entire globe, 98% of every human on this planet is vaccinated. But you've never heard about it in the US and Europe. And it's a public health policy not to vaccinate and allow you to get TB and treat with antibiotics. We got into using this drug because we thought it was going to be a simple pathway through the FDA, right? 100-year generic drug. So I have a lot of respect for everybody in this room who takes real drugs forward, because it's still a haul moving forward a 120-year-old drug. So these are some of the trials going on. We just had our international meeting in Athens, Greece. There's a total of 16 trials going on. The most advanced trials are using BCG for the reversal of new onset multiple sclerosis. In Italy, we're in phase 2 licensure trials at Mass General in type 1 diabetes, reversing established type 1 diabetes. Just so none of you missed the headlines, 2 weeks ago in the New York Times, there was a big report, again, of the health benefits of BCG. A Danish group showed that with 3 vaccinations in Europe that ceased about 50 years ago, but with public health records, if you got 3 vaccines and you were a European citizen, you lived 10 years longer. Now, why is that? Why is this old-fashioned vaccine changing morbidity and mortality? Well, that group had a little bit of bias because about 8 years ago, they went into the poorest part of Africa, Bandine, Guinea, and decided to really test whether BCG protected from tuberculosis. So they randomized about 200,000 infants, half BCG, half not. We're going to follow them fully for 12 years. They had to stop the trial after 3 years because in the low birth weight infants that didn't get BCG, 70% higher mortality. So it was those studies that made them reflect on really trackable health data in Europe of what does it do to everybody sitting here in the audience. So although I'm going to talk a little bit about type 1 diabetes, you're going to feel a little selfish as you hear this presentation and say, I want to have what they're having. This was a slide on the cover of Science. It was about the hygiene hypothesis, about how many of these diseases we're seeing in our society are the result of taking away the environment. I laughed at this picture because it was the milkmaids that got infected with the bovine tuberculosis, a virulent strain. So they were at the other end of the cow. So the picture is kind of on the right way, but not totally giving the right message. Okay, so what about BCG, and how did we get involved with doing BCG instead of a high-tech drug? Well, it was a number of years ago, we published a paper in JCI and a paper in Science saying, for the first time in mice, right, mice, you got to always have a word of caution when you're talking about mice. But when we induced TNF, or use the surrogate inducer BCG, we could even take end-stage diabetic mice and permanently reverse type 1 diabetes. So here was this 100-year-old generic drug that was reversing end-stage diabetes never seen before. In the case of the mice, and I always say these are mice, not humans, the pancreas massively regenerated and restored insulin levels. Whether that'll occur in humans or not, we don't know. Then the good news was everybody went out to their autoimmune mouse studies, and as somebody said, if you're a mouse, we got you covered. Whether you have lupus or diabetes or multiple sclerosis, BCG is highly efficacious. So we wanted to move this into human clinical trials, and probably in totally synergy with this trial. It's not really smart to go in clinical trials if you got mouse data, but you don't know the people have the same defects. So we spent a long time on biomarker development to prove people with long-standing type 1 diabetes had the bad T cells we could monitor, had too few of the good T cells, and other parameters of the disease progress. So one of the things we tracked were bad T cells. I don't want to show you too much data because everybody else has these glamorous presentations. But effectively, if you're a long-term diabetic, even 15 years out, we can find those bad T cells in there. But importantly, we could show the drug and culture could selectively and only remove the autoreactive T cells. So that became one of our, quote, biomarkers as we move this forward into human clinical trials with the supervision of the FDA. We also worked on the good T cells, which are all the rage. If I give a lecture over at Harvard Med School, it's always, what are the Tregs doing? What are the Tregs doing? They used to be called T suppressors, and then there was some data that wasn't quite very good, so they changed the name and they became fashionable again. So we can show BCG induces the Treg cells as well. So this is kind of our picture of how we view autoimmunity. You got too many bad cells in the infiltrate. You don't have enough good cells. And we think this 100-year-old drug is flipping that balance, as well as other people showing data now with a flip balance. But just so you put it in a context, everybody knows the rate of autoimmunity is on the rise globally, the rate of allergies. I mean, whoever grew up with a peanut allergy, okay? The rate of asthma is on the way up. And everybody knows it's the environment, and some people like to blame something toxic in the environment. We like to think about what's been taken away from the environment. And what's really obvious is if you don't have the right mixture of microorganisms, some of these diseases are going to surface. And the oldest organism out there, I'll show you a final slide, is Mycobacteria tuberculosis. So we think we have 100,000 years of evolution on our side as we march forward. This is some data. If you get tuberculosis, globally, you don't get type 1 diabetes.
It's amazing.
If you get a lot of vaccinations and you take away a lot of these diseases that you certainly don't want, you get all these other autoimmune diseases. And of course, if you get tuberculosis, you don't get multiple sclerosis. So highly protective, but you're not going to vaccinate people with TB. We've learned a lot of lessons. Talk about collaboration and people working together. The TB field was really the kind of the first group that showed those Goombas that people have in their lungs from tuberculosis are little factories of these good T suppressor cells. So it turns out BCG does the same thing as these little factories of tuberculosis. And so we went into human clinical trials. We did a phase 1 clinical trial, full FDA oversight. These were not investigator trials that are typical of academia. We followed people with long-term diabetes on the impact of 2 vaccines and followed them for 22 weeks. Remarkably, every single biomarker that we had perfected showed up a positive result. We didn't cure anybody, okay, but we saw the change in the biomarkers which were consistent of the drug eliminating the bad T cells and inducing the good T cells. This just shows some of the data. I see it's too geeky for this audience at this time, but we could see the death of T cells in the peripheral blood. We could also see the induction of TNF. It's known that BCG or tuberculosis induces TNF. We could also see the good Tregs. Importantly, and this is of interest to Kevin, we had spent almost 6 years trying to perfect whether the pancreas was turning back on because you can't measure insulin because the people are taking insulin. So you measure a surrogate called C-peptide. Remarkably, the assay out there for C-peptide was developed 40 years ago. The primary assay, sorry you guys if you're from Roche, was developed by Roche. It not only recognizes C-peptide, it recognizes proinsulin, so it's not very good, and had a lower cutoff level of 40. So we worked at perfecting our initial, probably not as good as Kevin can do for sure, but we perfected it to get down to these low levels. So we were very excited when we unblinded the data that we could see the pancreas, these are people 15, 20 years out, make some insulin after BCG. But the real surprise was when the data came in unblinded, the lower limit of the assay showed that even the control group at the start of the trial, as well as the treatment group, had remaining C-peptide. Remember what we've told everybody. I'm an endocrinologist. We told everybody that you get the disease, you go, you talk about doctors telling you things. You're going to get the disease, you're going to have this honeymoon, you're going to have a little bit of pancreas function for a year or so, and then your pancreas is dead. Here it was, by making the assay more sensitive, we were showing that people 15, 20 years out with this disease still had a live pancreas making insulin. Small amounts, but making insulin. So we've worked a lot on trying to perfect those assays. The top graph just shows you where the normal cutoff was for sensitive C-peptide assays. Can you guys see that? Okay, so here, where it was around 40 or so. So this is all people knew about, so it obviously looked like the disease stopped. But now we know the decay in the pancreas goes over decades, not just that one-time interval. And then as a humorous story, 2 years after this report, I'm in England at the European Diabetes meeting, and a British fellow who had just gotten his PhD done came running up to me, ready to hug me. Okay, now that's a paradox in and of itself. But the funny thing was, he said, Dr. Faustman, I have good news and I have bad news. And I said, well, what's the good news? And he says, well, I've been collecting urine for C-peptide in Scotland, Ireland, and Great Britain, looking for fat type 2 diabetics that have C-peptide issues. And instead, I kept finding C-peptide in kids with type 1 diabetes that had it for 15, 20 years. So this is really good. So I hug him back, and he goes, well, you want to hear the bad news? I said, okay, what's the bad news? Did I tell you the story before? Yeah. So what's the bad news? And he says, well, I got done with my thesis. I totally wrote it. I went before the review board, and the most senior person in all Great Britain sat there and said, got 2 damn bloody assays not working now. So there was like this resistance of the scientific community to believe the paradigm of this disease was just wrong. So this slide just shows you now a lot of people are finding C-peptide. So this is the textbook picture. You can find it in any medical textbook, the top one. New England Journal of Medicine, about 5 years ago, wrote a review article, the pancreas is dead within a year. And so now we've really changed that paradigm by developing better assays that the pancreas can live on for decades, not functionally, not fully functionally, but it's not quite dead. We also know these C-peptides, which is very important for getting FDA approval for these new things, correlates with the duration, but importantly, it correlates with hemoglobin A1C, the control of blood sugar. So a little bit of C-peptide is really valuable for keeping your diabetes under control. The trials that are going on with BCG are going on diverse autoimmune diseases. This data was published from Italy. And they looked for new onset MS. They, of course, did CAT scans, and they did as well as relapsing rate. And what they showed was BCG as 2 vaccines was better than the standard of care. The good news is the effect appears to be permanent. The bad news is it takes about 1 or 2 years for the systemic effect to show up on MRI scans. So that sets the stage really for all of us doing trials in allergy, diabetes, and other autoimmune diseases. We have 3 clinical programs going on at the Mass General trying to get licensure of this cheap, generic, safe drug for autoimmunity. We also got another very large donation from a person interested in fibromyalgia. To date, we're not as good as you, but we've raised $50 million nonprofit to get these trials moving. So it's a huge effort of the generosity of the public of wanting change in healthcare. Our current trials of phase 2, 6 vaccines fully enrolled. We're about a year into that 5-year period. As an example, we picked the toughest endpoint, so we're pretty serious, a change in hemoglobin A1C. Not to get too geeky, but we do epigenetics because the question comes up, why in multiple sclerosis, why for morbidity and mortality do you see such lasting effects of BCG? Well, it's now pretty obvious for those of us working in the tuberculosis community as well as with this vaccine. BCG permanently changes your methylation patterns of some of the most important T signature genes. So what we're able to now show is for the 6 most important signature genes for Treg cells, within 8 weeks after a vaccination, 2 vaccinations of BCG, we can see systemic changes in the methylation. So we think these kind of mechanisms, these are all the genes we track for methylation, are going to give us a clue of why these vaccines take a while to kick in, but then their effect lasts decades. That's more data. I don't want to bore you guys. More mRNA data, lots of data. Okay. So my point here is that old drugs can have really important purposes if you have the mechanism of action and you know biomarkers to track efficacy. So this has changed our view of, these are some of the first trials in type 1 diabetes, trying to reverse the disease when you have the disease. Up until now, all drug development, talk about frustration, has only been in new onset diabetics or pre-diabetics because they thought it was too hard to tackle people with end-stage disease. So we've, because of the generosity of the public, have decided to tackle the people who are most deserving of something affordable and maybe efficacious. People send me many slides, so one of the themes here is get dirty, okay? And dirt is good, so if you have kids, get them in the backyard, get that dirty dog your husband wanted, and make sure they get a lot of exposure to the environment. And last but not least, we really are all thinking, who work in this field, that we've got something really good, but it's really Mother Nature that has something good, because you probably heard the story 2 years ago, the paper was published in Science, that these spearlunkers in France were going down these caves, and they looked over and they saw this shriveled up guy, and they thought, oh, how unfortunate, somebody 70 years ago, 100 years ago fell down that cave. Well, he fell down that cave 90,000 years ago. So he was the best freeze-dried Neanderthal anybody had ever found. So everybody went after the DNA. We got to get the DNA out. We got to really see where our roots and origins are. Well, they went to sequence the DNA, and they kept sequencing and sequencing and sequencing. They kept coming up with one thing, mycobacteria. So there's no doubt that mycobacteria is the oldest co-evolutionary organism that set our genes and we set its genes. So with 90,000 years of evolution, we've gotten rid of genes we don't need because it regulates our genes, and it's gotten rid of genes that we regulate their genes. So now you've lost those genes, you remove mycobacteria from drinking water or from being a farmer in the soil, and all of a sudden you have a different reset of the immune system. So all these people seeing beneficial effects in trials are now really working on this hygiene hypothesis and trying to reintroduce this very, very old vaccine for many human conditions. So that's my story. Thanks for listening. Thank you for inviting me.
If you don't mind sitting down.
Oh, yeah.
Thank you very much.
Oh, okay.
So Denise, I found that your story is so interesting from so many levels. Number one, yesterday we actually on our Alzheimer's panel, some of the leaders in the world, started talking about microbiology and the same kind of areas that you're just describing as maybe being some of the genesis of Alzheimer's, which I found very fascinating. And we are invading our microbiome on so many levels. And so this talk and this panel, for me, vaccines and having something that's been proven for 100 years and then getting it approved to be a drug has been just a major struggle for you. Mm-hmm. To your right are three of the leaders in neurology in the world that are doing similar things, trying to get drugs through the FDA that in fact can help brain health, okay? And seeing biomarkers objectively in blood has been such a major challenge because of the blood-brain barrier. So there are biomarkers that are in the cerebral spinal fluid, unlike Henrik, which we still think that a spinal tap is somewhat invasive. If you can find these biomarkers that you thought were in the cerebral spinal fluid in blood very non-invasively, and you can team them up with the drugs and when you're trying to do trials and show more objectively that you're moving the markers that you're trying to move, or you're not moving the markers that you don't want to move, and you can do it non-invasively, you have a shot at revolutionizing a whole new way of getting drugs approved. And so I believe these three individuals are on the forefront of that. So I'd like to evolve now into a discussion around the drug approval process and some of the opportunities that you see for new neurodegenerative disease drugs, and what maybe the role biomarkers can play given this new level of sensitivity that we're able to see in blood. It's the equivalent, we said yesterday, of a grain of sand in 2,000 Olympic-sized swimming pools. That's what we can now see in blood. We can also differentiate a protein at a level of a single blade of grass in a field the size of Alaska. So like Steve Jobs did in innovating the iPhone, there has been innovation in a new tool that's been developed in the last several years that can do this in blood. And so these guys are trying to exploit that very rapidly because we've got patients that need our help today in neurodegenerative disease. Today they need this opportunity. And so now this is where the rubber starts to meet the road. So Al, I'd like to start with you. I'd like for you to provide your perspectives on how we can start to move into more objective approaches with biomarkers and linking them up to drugs and really change the game of getting drugs to patients sooner.
Thanks a lot for the question, and Nancy, I have to say that was a very inspiring talk. And you hit upon all the major issues, including early diagnosis, the way we measure disease progression, how subjective it is and how variable it is, and biomarkers, to your question, Kevin, helps us a great deal because, and I've been thinking about MS. You know, when I was training as a resident, we had no drugs for MS. And now we have about 14 drugs approved. And I was thinking, now what happened that created this revolution in MS treatments? And I have to say, if I had to pick one thing, it would have to be that we can measure the disease better using MRI as a biomarker. So we discovered years ago that you can do a 6-month study and count the number of MS lesions in the brain. And know whether or not a drug affects the rate of relapses. So MRI predicts relapse rate. And there's been a beautiful set of studies published by Maria Pia Sormani, a statistician, where she looked at all the drugs that have been looked at in MS, and the correlation between the effect on MRI and the effect on the clinical outcomes is beautiful. And so if I had to pick one thing, you know, we still don't have a great animal model for MS. And we still don't really understand fully how the disease begins or what causes MS. But the fact that we have an objective biomarker like MRI has really helped MS, and I'm hoping that for ALS, we can get something similar. Now, you mentioned blood, Kevin, and I think that there's a lot of excitement going on now with blood-based biomarkers. I was just talking with Jeff, and we have a poster. Actually, I think it's a slide presentation this week in Paris at the big MS meeting, that neurofilament light measured in serum is associated with response to treatments. So when people get on drugs that work in MS, the levels of neurofilament light fall dramatically. We actually did a study years ago in people who had the very first clinical event, optic neuritis or spinal cord disease that led to MS. That first blood draw at the time of the very first clinical event predicts their disease progression 20 years later. That's one of our posters this week. So, and then many of these neurodegenerative diseases are due to misfolded proteins. The accumulation of neurotoxic proteins, for example, for Alzheimer's, it's amyloid. For ALS, it could be TDP-43, and I think there's some really cool data that suggests that we can measure these abnormal proteins coming out of the brain into the blood, and but they're in small quantities, but because of the advancements in technology, boy, if we can measure that, because I think the key in many of these diseases is to treat early. And in Alzheimer's, I think we're going to be treating even before first symptoms one day. So if we could, but you can't do PET scans on everybody, which is the only way we can detect the abnormal accumulation of amyloid in the brain right now. But if we can detect it in blood, imagine what that would open up. So I'll stop talking because I'm hogging the whole session here.
So Al, you know, really appreciate those comments, and that's the breakthrough that's occurred in the last few years. And many of the leading researchers around the world that actually found NFL initially with these technologies are sitting in this room, and we had major debate and panels yesterday about a lot of that technology, and not just NFL, but tau and amyloid betas, and there's probably, I think, 10 or 12 different biomarkers now in blood that are very revealing, and it's allowing the whole pace to move so rapidly towards more objective FDA filings. Jeff, being with Lilly, I was curious if you looked at your career and where some of the biggest disappointments have been in getting drugs approved, And what role do you see now with these biomarkers from your perspective and how they're going to help us move this forward faster?
Yeah, well, thanks for inviting me to the panel, but speaking about disappointments is always troubling. And the way I like to think about it is, as we take every opportunity we can, and we take the best opportunities forward. And in each of those clinical trials, we learn something. So yesterday, I presented data from our c-McGase-STAT inhibitor. I presented data from our first base compound, where we were able to take stored samples and look at blood biomarkers and learn what could we learn from those old failed trials that we can apply to the new trials. And I think when we look at disappointing results, we have to be optimistic, and we have to say, what can we learn from them, and how can we advance the field forward? And what Alfred was saying, as far as where do we go next, I think, for me, it's all about disease understanding, and the biomarkers that we're developing and how we're using them. It advances our understanding of the disease. It advances our understanding of when things become elevated, how many years ahead of time from clinical symptoms. I think right now, for Alzheimer's disease, what it's telling us is we can see amyloid buildup so far in advance of clinical symptoms that maybe we need better clinical measures. Maybe we need things that can measure earlier so that you can see the first deviation from what normal aging would be. And I think that'll enable us then to treat earlier, maybe in the presence of actual symptoms, but maybe even earlier than that. And I think those are the disease understanding is the key for me to learn. The other part I wanted to bring up is repurposing. I mean, oftentimes in pharmaceutical discovery, we try things and they don't work, but we know a lot about them. And where they are safe, we have an opportunity to repurpose those things. And even in our preclinical efforts, and I know we have a Project ALS where we use some of our preclinical immuno-oncology medicines to provide those, or not medicines, they're preclinical drugs, investigational, in vitro investigational. But we provide those to researchers in academia to see if they have potential uses in ALS. And I think those public-private partnerships, and Lilly's a great opportunity, does a great job of providing that and creating that opportunity. And I think that's where we can help advance the field, whether we're struggling to get things through the regulatory process, or we just want to enhance the disease understanding. I think the more we interact with one another, the more we partner with one another to enrich the disease understanding, I think is going to be the key. Now, biomarkers, we were talking about this also before. I don't necessarily see biomarkers as the key to regulatory approval, but I see biomarkers the key to making really important decisions in clinical development. Is the molecule safe? And then are you hitting the target? And then when you hit the target at the appropriate levels, are you able to see a response in a pathway that you understand? And so target engagement pharmacodynamics allows you to pick a dose to advance in to a phase 3 clinical trial where you could have an FDA approvable endpoint. And I think those things are where biomarkers can really help us.
It's excellent. And Ramon, you've been participating with us across so many different aspects yesterday of concussions actually being potentially an environmental factor to lead to CTE, to potentially lead to Alzheimer's. We heard from Hendrik, we heard from Bob Stearns, we heard from Ann McKee describing the severity of the actual ultimate endgame traumas. You've been watching these biomarkers evolving over the last 7-8 years. What is your view of the potential to actually see in blood these diseases so much earlier to enable treatments to occur earlier? And I'm thinking primarily of the Alzheimer cascades that we heard from yesterday, from Rudy and others. Do you feel, as a clinician, and the ability to actually create diagnostics in this area, do you feel that there is going to be opportunity to see disease so early in this neurodegenerative landscape that we will be able to get treatments so much earlier that are actually going to lead to either reversing or at least keeping the disease where it is long before symptoms ever present?
Well, thanks so much for the question and for, again, the invitation for this meeting, which has been very, very stimulating. I mean, gosh, I think obviously early diagnosis is a very attractive feature, certainly makes a lot of sense. And in cancer and many other conditions, I think has proven to be the case, and I think likely is going to be the case in neurodegenerative disease as well. On the other hand, that's only the case if we have a very effective therapy that can be given pre-symptomatically, and also a very, very safe therapy that can be given asymptomatically. So of course, I do think that that is going to be a role for biomarkers. On the other hand, I don't think that early diagnosis and early therapy is the only solution, right? And there are certainly going to be many conditions where probably the wisest thing to do is watch and follow the condition until you know when the therapy, when the cost-benefit analysis, when the cost-benefit ratio of the therapy is going to be worth it. I think the bigger role for biomarkers, which is also we have talked about here, is appreciating the complexity and the heterogeneity of a lot of the diseases that we deal with. I think all of us were trained in the reductionist pattern of biology where we think that everything can be reduced to understanding genes and understanding molecules, and that is certainly the principle, but we have greatly underestimated how complex it really is. I recall when I was an assistant professor and I told my chairman in our yearly counseling meetings that I wanted to study traumatic brain injury, and he told me, Ramon, you are crazy. Traumatic brain injury is way too heterogeneous, way too complex. You will never understand it. You should study something simple like Alzheimer's or ALS. And in truth, I looked around and I knew something about Alzheimer's and ALS, and I realized that they weren't simple either. On the other hand, the folks that were studying had kind of put some blinders on, had assumed that it was very simple, had assumed that it was an amyloid disease or a SOD1 disease or what have you, and that was wrong. I think all these common diseases are going to be very heterogeneous, and they're going to have common, and they do have common phenotypic expressions, but the pathophysiology is mixed, and inevitably, I'm convinced it's going to be the case in TBI, but I'm also convinced it's going to be the case in ALS and in Alzheimer's and everything else. We're going to need combination therapies, right? We need combination therapies for everything. Cancer is usually treated with combination therapy. Tuberculosis is treated with combination therapy. And when you're dealing with a complex disease, a component of what's going on in Alzheimer's and ALS and TBI is going to be related to protein aggregation problems. But another component is going to be inflammation, and another component is potentially going to be microvascular pathology, and different patients are going to have different mixtures of these heterogeneous pathologies to different degrees, and the biomarkers are going to be essential in order to assess how much of one particular endophenotype, as it were, is active in that particular patient, and therefore what therapies this particular patient needs, right? So when a cardiologist is evaluating a patient with coronary artery disease, they don't just give them a drug for coronary artery disease. What they do is they measure the blood pressure, and if there's hypertension, they treat the hypertension. They measure the cholesterol level, and if the cholesterol is high, they put them on a statin. They measure inflammation with CRP, and based on that, they may put them on an aspirin. And that's what it's going to take, I think, to deal with all of these diseases, including some that we've been assuming are simple when they really aren't.
Very good. And you know, obviously, Nancy went through a list of a lot of neurodegenerative diseases when she was actually in the room for her diagnosis with Pete, and they kept going up the list. And you were trying to put a lot of that in perspective. And Denise, you actually proactively got in there and found something that could be used, and you've pushed it through the FDA. I was wondering if I can close on this discussion. Two of the largest drug companies in the world being Lilly and Biogen. I wondered if Al and Jeff could just describe, in the next 5 years, as you look at your pipelines and you look at the opportunity that we see ahead of us, where do you think we can materially transform the ability to treat neurodegenerative disease in the next 5 years, realistically? And paint that vision for us to give us a little bit of hope around how these treatments have a shot of really changing the world. Because you guys are investing billions into this category. So Al, I'd love to get your views.
So I do believe that we're on the cusp of new treatments. I mentioned MS, but recently we got a drug approved for spinal muscular atrophy, which was using an intrathecal antisense oligonucleotide that we, in collaboration with Ionis Pharmaceuticals, we injected into the lumbar cistern, into the spinal fluid. And the drug was approved by FDA in record time, like 3 months after submission, mainly because they were so compelled by the data. And there's some lessons learned there. First of all, we're doing the same technology for ALS. We're actually doing what you said, which is taking what is probably a heterogeneous disease like ALS. I mean, the phenotype at the end of the day is similar, right? That's why we recognize it as ALS as clinicians. But we know that it can be caused by SOD, C9orf, and a whole variety of other upstream genetic causes. So what we're trying to do is divide ALS into these subsets. And we have we have an antisense for SOD, we have an antisense for C9, and so forth. So we're trying to make the populations homogeneous, and use this technology. In the case of SMA, what we've learned is that the drug, even though you inject it into the lower back, goes all the way up to the brain. It causes a change in gene expression, which is what we wanted to do in SMA. We learned. We had 3 registrational studies. We did a study in infants, in children, and in pre-symptomatic newborns. In the case of SMA, we had the advantage that you can test somebody for their genetics, and you know that they're going to have SMA, because if they have a homozygous deletion of SMN1, they have SMA by definition. It was a very difficult trial to do, because how do you find them before they're symptomatic? In Taiwan, they were doing newborn screening for SMA. So we found some patients in Taiwan, and in the States, we were able to get some, and in Europe, we were able to get some patients who were siblings of affected children. What we found in these studies, just to make a long story short, is that the drug was much more effective if you start early. If you start in the newborns, the pre-symptomatic newborns, many of these children, even though they have type 1, and most type 1 children die by age 2. Not only are they not dying, they're actually gaining motor milestones, most of them on time. So, and then as you go later and later, the efficacy of the drug, it still works, but not quite as well, not quite as dramatically. Even in the infant study, if we divide the short disease duration with the longer disease duration, the effects are different. So what's the equivalent then of the genetic test? For all these other diseases, I think if I go back to biomarkers, I look forward, to a day when our iPhones, the great advantage of iPhones and all these digital tools is that they actually are interfaces with our brain through our eyes and our fingers. So, and they're very good at detecting movement, which is one of the first things that get affected in ALS. And also, they're probably going to be very good at detecting when you're not thinking properly or where your memory is going wrong. And it's going to there's going to be an alert that says you should go to your doctor because something is wrong. And then the doctor hopefully will draw blood, and we'll be able to figure out what in a pre-symptomatic stage, what the disease you're destined to get is. And then we often think of these diseases as Alzheimer's or Parkinson's. Actually, the truth of it is when people go to the doctor, often it's very complicated. Often they have more than one protein that's accumulating. Sometimes it's both alpha-synuclein and amyloid. So you get diseases like diffuse dementia with Lewy bodies. So we call it these names, but ultimately I think we're going to move to a molecular diagnosis based on the abnormal protein that's building up. Even in ALS, we now know, we used to think of ALS as a purely motor neuron disease. Now we know that half the patients have frontal temporal dementia. And so these are not, these diseases are much more complex than we make them out to be. But ultimately, and then instead of naming these diseases, we're going to figure out what the molecular problem is based on biomarkers, and we're going to take the drug off the shelf that fits with that abnormality, and we're going to treat, I think, before symptoms.
So Al, that brings such hope to the movement of what we see. I feel like this group that has assembled in a collaboration, it's occurring across all these different disease states on biomarkers, has revealed more than anything else is the opportunity that we can do this is already here. These technologies have been around for 3 years now, and the urgency of how do we break down and get this further infused and further understood by the world so that every scientist has a shot at transforming the way medicine's practiced with just getting in there and understanding the accumulation of proteins at a molecular level already can be done. And so you guys, you're utilizing the tools, I know. Jeff, you guys are utilizing the tools, and getting to these diseases earlier is the other byproduct of these tools because they can see disease happening long before there's symptoms. That combination of seeing it early, then having treatments, what do you think we're going to see in the next 5 years coming out of Lilly that's going to really shake up the opportunity to put some of these diseases behind us?
Yeah, and it's a great question, and I don't know that coming out of Lilly, is the exact example, but I think the antisense example, where you have a therapy approved in record time by the FDA, opens up an opportunity across multiple genetic diseases. And I think that's an excellent example of a breakthrough innovation. And whether that's going to come out of Lilly and the Lilly portfolio, I don't know. But those examples need to be acted upon. And I think those are just the true innovations where you actually can achieve an approvable medicine using antisense. I think it's a great opportunity. And we were talking about this last night. When are biomarkers going to become valuable? It's going to be as soon as we have that first therapy that shows an effect. Because then everything is going to fall into place. Whether that's coming from Lilly, Biogen, or another company, I think that is going to be a huge breakthrough. And I think as soon as we have the first medicine in Alzheimer's disease that shows a therapeutic benefit in an approvable trial, I think we're going to have an opportunity to start assembling all of the biomarker data that we have with all of these ultrasensitive tests and really build that understanding of what's driving that effect. How early do we need to go? And it'll all fall into place. And the reality is we don't know when or where it's going to come. But I think we all, every time a trial's nearing its endpoint, we all sit on the edge of our seats and we hope. And as a biomarker scientist, you hope because you really want to understand that disease, because it's not always the first one that's the best one, but it's the first one that gives you that understanding of how to make a better one. And I think that's where we can continue to be iterative, we continue to advance, and then we continue to go wide. You get a first indication in one disease, you have opportunities then to learn from that and learn the similarities. So the spinal muscular atrophy is a great example there too. You learn from one disease, how can you apply that to another one? I think that's going to be the big breakthrough. And I think we can take advantage of, which I mentioned already, the immuno-oncology, the autoimmune. There's people developing therapies in diseases that aren't Alzheimer's disease, but maybe they are related, and maybe they will have a benefit. And how do we take advantage of that? When is the right one? What is the right time? And then when can we advance it? And I think, those, you hate to call it serendipity, but, if you're paying attention and you know that these things interact, reacting to that first key bit of data is going to be the thing that you want to do, the breakthrough. Recognize the breakthrough and advance on it. And I think that's how we're going to, really see innovation happen, really make a difference for patients over the next 5 years.
So Jeff, I think we calculated there's about 15,000 scientists that are linking in with these videos through, thank you, Steve Jobs, for what you've done with the internet. There's innovators all over the world that are responding to what happened yesterday. And a lot of this has become social. And one of the things that I found very intriguing was a description from one of the leading pathologists in the world yesterday described it. This is like a blind man on the elephant, because we've had oncology, we've had neurology, we've had all different types of disease states going across the blood-brain barrier, and they're all watching these blood biomarkers being presented at their deepest levels. In the afternoons, we get to the very deep levels of the biomarkers, and they're saying, we're seeing things that are going to lead to breakthroughs that we didn't even think about before. They're collaborating across these disease states, and the biomarkers that are circulating in the 60,000 miles of blood are actually there. It's almost like the islands. You look down at, from up above, and you look down and you see these islands, and you don't think they're attached. But if you drained all the water, they're all connected. And biomarkers are there, and it's just now we're able to see them with these tools. And I think Nancy said it the best to me, and I'll never forget this part of the discussion, Nancy. You said that Pete had a new drug put in him, and it might have become the worst day of your life, was when the hope was taken away from that particular drug working. He had so much hope and inspiration for this particular drug working. Thank God for the Lillys, thank God for the Biogens that are deeply in there trying to innovate. The drug went in him, and it probably was 3-6 months later in the trial, they found out that that particular one was not going to work. And that means you've taken this hope for those that really need it, and you just take it away on that day. But thank God you're continuing to just pound at it, because these drugs are needed, and the way in which you're going at it with biomarker assessments are going to allow it to be revolutionized. So one of the things we feel has been such a passionate part of what we're all about is the funding. We heard 50 million discussed by Denise. We heard 220 million discussed by Nancy. I want to, first of all, thank this panel and what you do. Your heroes continue to plow away. There's a lot of people watching. There's going to be interactions that are going to occur. The panel session this afternoon is going to be going after cancer, and pancreatic cancer is the first one that we think we have in our sights that maybe we can put it in the rearview mirror. We're going to talk about that. So I want to thank the panel, but I'm going to ask Nancy to stay on the stage because we'd like to just talk a little bit more about funding. So thank you very much. Denise, thank you so much.
Okay.
You were awesome. I know, no, you weren't geeky at all. So Nancy, let's see how we do this.
Can I just add a couple of things? And of course, again, I keep reminding everybody I'm the layperson here, but I'm the mother here, so which gives me a lot of credibility. Couple of things in the 6 years that I've been thrust. Now, I was an entrepreneur. I was a business person. I was a marketing person. I know I failed biology, so I know that. So let's, because when I was going to college, I knew that wasn't where I was going. But in the last 6 years, I have the war wounds to show I've been in the trenches. A couple of things that were said here that I would like to bring up. The one is hearing the drug companies talk about their closets full of drugs. And as a mother of a person with a disease that's looking for a treatment, there's got to be a way to get those drugs out of the closet and be able to get them to every single person in here who's studying a specific cause. You know, that was probably one of the hardest things for me to put my brain around when I came into this space was the lack of collaboration. You know, I love the analogy you just gave with the islands, because in the 6 years, I've seen the water going down. They were like icebergs when I came in here, especially in our space, and I see that happening. But why isn't there a negative results database? Why isn't there something? We have these drugs, you know the mechanisms, you're the smart people, that's why you went to school all these years. You understand what mechanisms go with what drugs and what might be for certain diseases. That's something. But in my world, in the patient advocacy world, urgency is the most important word that you can use all day long here. And the end game is to get the drug to the patient. And one thing that the Ice Bucket Challenge money has gone to, which I am so proud of, is a collaborative effort between patient advocates, researchers, clinicians, physicians to create an ALS FDA guidance document. And we have been told by the FDA that this guidance document is a gold standard in disease spaces. So that's another piece to help push these things through the FDA to get them to market as quickly as possible. So I would urge people in disease spaces to look at what we did in ALS and produce these guidance documents so that they can get through the process a little bit easier.
It's well stated, Nancy. You know, and for me, we've got such a burden because there has been a tool invented over the last several years that can truly see inside the body and do things that is revolutionary. And we've got to find a way, it's a burden on all of the people in this room to find a way to break through very rapidly to get these tools applied to this opportunity that's sitting in front of us. And I know the FDA, in fact, several of the people here in this room, we went into Washington, D.C., was it last week, Dan? We had a whole room full of the FDA. We were describing to them, and they were sitting there like, you got to come back. This could be the most revolutionary opportunity to truly be able to move an understanding of what's going on in the body that may be completely unrelated to the way we define it today. You've got pancreatic cancer. You've got brain cancer. You've got lung cancer. No, you've got molecules circulating that are stimulating cell growth at a rapid pace that actually is the trigger no matter where the cancer is. But we got to change the paradigm. We got to look at this very differently by removing the water and seeing how they interconnect. And it requires collaboration of the biggest minds. My big fear was, can we get these big minds, these are the people that are like the top in the world, to come to something like this, to start collaborating on a tool that really has a chance to achieve this. These guys are coming, and they're the biggest minds, and the people that are watching this from outside can see this opportunity. So you bring an urgency into this is exactly what we need, and this is what we're praying we continue to apply it and let the big minds really now apply these tools. Funding is what you've been able to do. Bob and I wanted to do just a small token of our appreciation. I'm going to ask Bob to come to the stage. Bring it. So.
You know, it's funny, I use the word disruption a lot because that's what we did. We disrupted, and that's what the mindset I think everybody in here is doing. The other piece is, you know what this is? This shows what storytelling can do. One thing Pete was very, you know, for 27, he was wise beyond his years. He knew he had a compelling story to tell, and he wanted everybody to hear the story. He wanted everybody to see his progress. But the other thing that he did, I don't know if Al is still here, but he wanted to get in, and as I said at the end of my speech, bring your patients into your boardrooms. Bring your patients into your labs. Bring your patients into your conversations. Because guess what? Everybody in here has a story. No matter where you are on what profession, what industry, whatever you're in. We all have a story. And as Bob said, you all have someone in your lives, and Kevin, that is having a struggle, a challenge, whatever it might be. But it's the story of that person. And we in ALS believe that that was what kept us in the shadows for so long, that people, we couldn't communicate, right? Technology allowed our patient community to communicate where for so long they were silent. But when the stories started being told, the progress started being made, the disruption happened. This was based on those stories.
The spirit of what you've done, and you are an angel, Pete's an angel, of spreading the spirit around the world through something as simple as an ice bucket challenge. We have a small check that we'd like to donate to. This is on behalf of the Powering Precision Health movement, as well as the Massachusetts Biotech Council. We would like also challenge everybody in this room to take the challenge and to spread this around the world again. Just keep it going. So we're all challenging you, Bob and I.
Well, before I get wet.
I know. I know. That's the one thing.
Yeah, we're definitely beforehand.
So, you know, talking about funding, which is what the root of all of us are, right? Because we know that it's the money that's going to drive all this. Philanthropy was totally turned on its head. You know, instead of going after that big fish for, a couple of months and then asking for the $1,000 check, we would raise it in about 10 seconds with $5 and $10 donations. But if you look at even the broader scope of that, it's the age group that gave. And that's my final word to everybody here. This is good news for every single person in here, in nonprofits, in disease spaces, is that my son helped motorize a movement that got that selfie generation to dip their toes in the waters of philanthropy, and that's going to help us all.
Oh, here we go.
Oh my God.
Without any further ado, this is because you looked adversity
There's
in the
real
eyes.
ice in here.
Yeah, oh Bob, you're going to be cold. But wait a minute, I'm moving over here.
I'm ready. You ready?
1, 2, 3.
Thank you. Thank you to everybody, and thank you for everything that you do. Keep up the good work. We're all counting on you. Thank you. Oh, thank you. You are so wet and cold.